Memory formation is known to be one of the most important studies in neuroscience. By studying brain activity, scientists are able to gain a better understanding of how the brain functions in processing information and forming memories. The research paper for discussion is from 2011, titled Preplay of Future Place Cell Sequences by Hippocampal Cellular Assemblies, and is by scientists Dragoi and Tonegawa. The study aimed to investigate the brain processes involved in memory formation within mice. In particular, it focused on the neuronal activity occurring in the cells of the hippocampus, known as place cells. The firing activity occurring in the place cells of the 4 mice was measured using electrodes. The mice were presented with different spatial locations in order to observe whether their place cells formed memories. Preceding their first experience on linear tracks, neuronal activity was recorded in the mice during rest sessions in the sleep box. Following familiarization with the linear track, the mice underwent a recording session, with the recordings continuing while the mice for the first time explored an L-shaped running track, in which the familiar and unfamiliar arm were made contiguous. It was revealed in recording the firing patterns of the mice that replay was present. This was indicated when the firing patterns associated with the mice running along the track were replayed during their moments of rest. In exposing the mice to a new spatial location, results showed that even before gaining access into the new arm of the track, the firing patterns recorded were similar to that of those recorded on the first arm. This phenomenon was termed preplay. The proportion of preplay events exceeded the chance level of 3.2%, indicated by the dotted line. This exhibits evidential proof of preplay having a factual basis. In comparing the recorded events, it was observed that replay firing occurred about 3 times more frequently than preplay. The neuronal activity associated with replay revealed that the mice were reinforcing their knowledge of spatial location by replaying information acquired during their rest breaks. It's believed that replay consolidates memory. In addition to this, it was discovered that neurons have a preplay mechanism, enabling them to rely on prior experiences to handle the challenge of a new activity or spatial location. A more recent research paper from 2013 by scientists Dragoi and Tonegawa, titled Distinct Preplay of Multiple Novel Spatial Experiences in the Rat, further investigates this phenomenon known as preplay. The methods and materials were similar to that of the discussed report, although the updated research report used rats rather than mice and focused on gaining a more in-depth understanding of preplay. The findings of the 2 experiments were alike in confirming the presence of preplay. However, the updated report included a more thorough results section regarding the preplay phenomenon. These studies exhibit the importance preplay has for the brain to make preparations for new locations. Neuronal activity revealed evidence of preplay, which suggests that the hippocampus plays a significant role in the development of spatial memory. With future research, a more comprehensive understanding could be achieved regarding the processing of location and memory within the brain.